In the previous session, we were discussing about the cell structure and its organization, and we were discussed cell organelles, few of them mitochondria, endoplasmic reticulum, Golgi complex, and so on. So let us today continue with peroxisomes and lysosomes. They are peroxisomes are single membered in enclosing a granular matrix, and they contain catalase and peroxidase enzymes. These catalase and peroxidase enzymes are going to destroy the peroxides and free radicals generated in the cell during metabolic process, which are going to have a harm effect on the cells, and these peroxides and free radicals are going to be nullified by the action of peroxisomes or peroxidases and catalases present in them, thus protecting the cell. Coming to the lysosomes, lysosomes are also called as suicidal bags of the cell because they contain hydrolytic enzymes or hydrolyses which are going to break the molecules, and these lysosomes are single membraned vesicle structures and having very low pH of up to pH 5, and this is going to uh, kill the bacteria which are going to enter the cell and thereby protecting the cell. And it is also called as suicidal bags of the cell because if the cell is in a harsh condition or unfavorable condition, it is going to release these cytolytic enzymes and kill itself. It is going to kill itself by releasing these enzymes, hence it is called as suicidal bags of the cell. So continuing, we have to also discuss about the nucleus and the central part of the cell nucleus. So cell membrane, the outermost, followed by the next cytoplasm and its cell organelles, cytoplasmic organelles, and the central part of the cell, which is called as nucleus. This nucleus is also called as control cell of the cell. It is present in all cells except matured RBCs. So matured RBC doesn't have nucleus. The structure is made up of two membranes, outer membrane and inner membrane, and this outer membrane is continuous, forming a endoplasmic reticulum like smooth and rough endoplasmic reticulum, whereas the inner membrane forms a numerous small pores called as nuclear pores where the movement of RNA molecules takes place. And the function of nucleus is to carry out the genetic material, and it contains DNA, and a process such as replication, transcription, RNA processing, ribosome synthesis takes place in this nucleus. So this is about the nucleus or cell uh, organelles completed with nucleus. Now we are going to continue with chapters or unit like carbohydrates or biomolecules. Before going to that, one should know the basic of chemistry or biochemistry, that is the chemical groups, valencies, and biopolymers and its bonds and enzymes. So this is basic you should know. So like in you have in a language we will start with alphabets. Similarly in chemistry we starts with these basic informations. So what is chemical group? Two or more atoms bound together as a single unit and forming a molecule. So there are different types of group are there in chemistry, and they are as follows: aldehyde, its formula is CHO; ketone, its formula is CO; and alcohol OH; and esters COO; and ether O. In this the line represents attaching to alkyl or aryl group, where it is going to represent it as R dash CHO for aldehyde and so on. So the bond are going to be attached with alkyl groups, and amine is a NH2 group. So these basic chemical groups formulas and its name should be understood for a better understanding of biochemistry. Coming to the Biopolymers. Before going to biopolymers, we have to discuss about valency. So valency is the combining capacity of an atom in known as its valency. The number of bonds that an atom can form as a part of compound is expressed by valency of element. So how atom is going to attach with another, and how much bondings it is going to have, that is represented by valency. Here there is a representation. In the first column we are having name of the element, and second column symbol with the valency. In the third column, hydrogen symbol H and its valency is 1. It is going to form a bonding with another atom with a single bond, whereas oxygen with 2, and phosphorus with 3, and carbon with 4. So whenever you make a structure, or whenever you write a chemical structure, you should keep in mind this should be satisfied, and bond should be according to the valency shown in this. So hydrogen you should show with 1, oxygen with 2. So that is the proper way of representation. And next is we have to go about we have to study about biopolymers or molecules. In biochemistry, there are so many biomolecules like carbohydrates, proteins, lipids, and nucleic acids. They are polymers and not vitamins and minerals. These are polymer forming biomolecules: carbohydrate, proteins, and lipids, and nucleic acids. Here four columns are there. The first column represents the polymer formed by the biomolecule, and the second one the building block of these polymers, and third one the bonding occurs to form the polymers, and the fourth one the enzyme which is able to going to cut these polymers or digest these polymers. The first one is carbohydrate. The carbohydrates are having building blocks monosaccharides, and the bonding seen in carbohydrates are glycosidic bonds, and the enzyme which are going to break these glycosidic bonds are glycosidases. And the second protein, it is having the building blocks amino acids, and these amino acids are going to be linked by peptide bond, and the enzymes which are going to break these peptide bonds are peptidases. And the third biomolecule is lipid made up of glycerol and fatty acids, and bonding is the ester bonding, and the enzymes which are going to digest these lipids are esterases or lipases. And the nucleic acids having building blocks made up of nucleotides, each nucleotide attached to another nucleotide by phosphodiester bonds, and these nucleic acids are going to be cleaved by phosphodiesterase enzymes. So this is a basic biochemistry. These enzymes name should be understood and bonding should be understood. So that for digestion process, what type of enzyme is involved in digestion of carbohydrate and protein, you can be easily able to understood. And then first column and the last column you should refer for digestion chapter. For digestion of carbohydrates, proteins, or lipids, you have to refer first column and the last column. That will give you an idea about digestion of these molecules. So let us continue. This is about basic of biochemistry you should know. Now we are going to start with carbohydrates. The first biomolecules which we are going to discuss is carbohydrate. Carbohydrates, as the name suggests, carbon, hydrogen, and oxygen. Carbon, hydrate means OH. So usually, generally, if you in a simple way, if you want to tell you, they are made up of carbon, hydrogen, and oxygen. They are the most abundant biomolecule in the nature. They are present widely in the plant and animal kingdom. And the definition for carbohydrates are is it is defined as polyhydroxyaldehydes or ketone or compounds which give these on hydrolysis. So you can see here. So polyhydroxy means many. Poly means many. Hydroxy means OH, and aldehyde and or ketone. Aldehyde or ketone we have just shown in the chemical groups. So aldehyde means CHO and ketone means CO group. So this you should keep in mind whenever you study a definition, what it explains and whether it is present in the structure, or it is going to be justified by the structure, or compounds which give this on hydrolysis. So you can see according to the definition there is a structure. you can see two structures has been written. The left side is the glucose and the right side is the fructose. The left side glucose is having OH groups, so underlined is OH group. So how many OH groups are there? You can see 5 OH groups. So from the second carbon atom, second, third, fourth, fifth, and sixth, and that's why it is called as poly many OH group hydroxy. And in the first carbon group, CHO is there already in the glucose. That is the first left side compound, and the right side compound is fructose, where you there also you can see OH groups in the first carbon atom, third, fourth, fifth, and sixth. So it is also polyhydroxy, but here it is not already. It's a ketone CO group. So the right side one is fructose. So glucose or fructose both are carbohydrates, and they justify the definition explained as explained. So this is about definition, and with example glucose, fructose, lactose, maltose, sucrose, starch, cellulose. There are so many carbohydrates are there that we are going to study along with classification. So now we are going to study classification of carbohydrates. Carbohydrates are classified based on the monomeric units, how many monomeric units it contains. So if a carbohydrate is made up of a single sugar unit, then it is called as monosaccharide. Mono means single. Saccharide. Sharkara means it's a, usually people tell it's a derived from a Sanskrit word. Sharkara means it's usually we have studied in a Sanskrit word. It refers to sugar unit. So mono means single. So single sugar unit. Disaccharide means made up of two sugar units. And oligosaccharide. Oligo means few, means made up of 3 to 10 sugar units. Poly means many, means made up of more than 10 sugar units. So single means mono, 2 means di, and 3 to 10 is oligo, and more than 10 sugar unit is polysaccharide. So this is about the classification of carbohydrates based on number of monomeric or sugar units. Coming to the detailed study of this, the monosaccharide, which is the simplest carbohydrate, example glucose and fructose, as shown in the definition. It, in that example was written glucose and fructose. They are monosaccharides, and these monosaccharides cannot be further hydrolyzed to smaller units because they themselves itself is a smaller unit. You cannot convert them into further smaller units. For example, the value of the lowest value of a money is now is 1 rupee. So if 1 rupee you are having, that is a monosaccharide. So if you from 1 rupee you cannot get another change. Now 50 paisa and 25 paisa is not valid. So 1 rupee itself is the simplest form of the rupee. Similarly, if it is a 10 rupee, if you give 10 rupee note, you can make 10 rupee into 10 1 rupee coins. 1 rupee, 10 coins you can make. So that is like polysaccharides or oligosaccharide. So like that, the simplest form of a sugar or a carbohydrate is a monosaccharide, which is sweet in taste and soluble in water. These are the properties. Based on the function group, these monosaccharides are classified into uh, based on function group. They are classified into aldoses. If a carbohydrate contains the CH group as a function group, then it is called as aldoses, and if its CO group is present, then it is called as ketoses. So aldoses example is glucose as shown in the figure where CHO is present. Ketosis example is fructose where CO is present. This is a classification of monosaccharide based on function group, whereas based on number of carbon atoms, you can also classify the monosaccharide into trioses, tetroses, pentoses, hexoses. 3 carbon means triose, 4 tetroses, 5 pentoses, 5 example pentoses ribose, 6 hexoses example glucose, fructose, and so on. So this is about a classification of carbohydrates in that monosaccharide. In that monosaccharide, further classification based on two aspects. One is function group and another one is number of carbon atoms. Now we are going to discuss about the next carbohydrate, the second one, that is disaccharide. Di means 2, so contains 2 monosaccharides, units linked by glycosidic linkage. Here 2 sugar units are there. For example, sucrose made up of 2 sugar units means glucose and fructose, whereas maltose made up of glucose and glucose, lactose made up of glucose and galactose. Sucrose present in sugarcane, maltose present in wheat and barley, and lactose present in milk. So this is about disaccharide. Next one is oligosaccharide. Oligosaccharide, as explained, it is made up of disaccharide ended with the 2 sugar unit. So next one is 3 sugar units. So 3 to 10 sugar units or monomeric units is oligosaccharide. Here also they are linked by glycosidic linkage. Example for oligosaccharide is raffinose made up of glucose, fructose and galactose. These 3 sugars forms raffinose. And polysaccharide, this oligosaccharide ended with the 10 monomeric units. So more than 10 is the polysaccharides. So polysaccharides means made up of more than 10 sugar units. So they here also they are linked by glycosidic linkages. Example, starch, cellulose, glycogen, heparin, hyaluronic acid. So these are the example for polysaccharides. In the polysaccharides, we can further classify them based on the type of sugar units present, same type or different type, as the homopolysaccharide and heteropolysaccharide. Since a polysaccharide made up of more than 10 sugar units, if all the 10 sugar units, more than 10 sugar units, all these are made up of same type of sugar units or monosaccharides, then it is called as homopolysaccharide. You can see in the left side example, starch, glycogen, cellulose and inulin. It is inulin, not insulin. Inulin is a homopolysaccharide. And right side you can see if the more than 10 sugar units present in the polysaccharides are not of same type, they are of different type. Hetero means different, homo means same. So hetero, different type of sugar units made up of more than one type of monosaccharide, they are called as heteropolysaccharide. Example is agar, hyaluronic acid, heparin and keratin sulfate. So this is about classification of carbohydrates based on number of sugar units. In that, few of these classification contain subclassifications as we discussed. So further we are going to continue in the next session. Thank you very much.